It's my pleasure to present some of our recent work to our B.I.G students. And I really appreciate that many students keep their cameras on so I can see their reactions. So that can make this session more interactive. So yeah, so my lab has a focus of developing computational and statistical methods for analyzing single-cell RNA-seq data. So as you know that we previously had RNA-seq technologies, and I think about 5 or 7 years ago, this technology has been advanced to study single cells, which means that you can measure the transcriptome within each cell. And this new type of data poses a lot of opportunities, but also computational challenges. So for today, I'm going to introduce our 2 recent papers in this area. So the first method is called PseudotimeDE. As the name stands, we want to do differential expression analysis, short as DE, under the context of cell pseudotime. So I will introduce what it is about in detail later. And this paper was recently published in Genome Biology. And you can check out the full text of the paper. And this was led by my PhD student who is second year in the bioinformatics PhD program, and Dongyuan Song. So that's the first work. The second work is more computational, even more computational. So we develop a simulator that can generate single-cell RNA-seq data and other protocols, not just RNA-seq, but other types of single-cell gene expression count data. And the advantage of our simulator is that it can preserve gene correlations so that the simulated data are more realistic. So this paper was accepted by the RECOM conference, and it was recently out at Genome Biology. Sorry that my slides hasn't been updated, but it's already out. And you can also check the full length of the paper, by searching the title. Okay, so I'll first introduce PseudotimeDE. Given our time limit, and also I understand that our audience are undergraduate students, so I will try to leave out some technical details, but focusing on the big picture. So what is pseudotime inference? So in short, or in simple language, it is that we want to order cells by their similarity of transcriptomes. So we know that when we measure a single cell RNA-seq data set, we measure a batch of cells simultaneously, and we don't know their temporal order in some differential process or some immune process. We have no temporal information at all. However, we hope to reinstall that information by ordering cells in some way. So let's just look at this picture. Here, each dot is a cell, and we're basically showing those cells by projecting their gene expression levels. Each cell may have about, I would say 20,000 genes expression levels for human, right? So that's a really high dimensional space. We cannot visualize a 20,000 dimensional space. So instead, we use some dimensionality reduction techniques to project the cells to a 2-dimensional space as this, right? And then we see, oh, by eyeballing, we can see there's some trend. And indeed, we use a computational approach to find the trajectories like those. And then let's say that we can find those 2 trajectories, then we project every cell to its closest trajectory. And the projection coordinate, after the projection, these coordinates along each trajectory is called pseudotime. So the reason we call it pseudotime is because it's not real time. It's sometime we inferred from the gene expression data. So popular methods for this task, pseudotime inference, include Monaco 3, T-scan, and Slingshot. And here the pictures I'm showing here are produced by Slingshot. So for technical people, I can briefly say that this is the principal component space, PC space. Within the space, the Slingshot first does this is called minimum spanning tree, an algorithm to find this tree, and then you try to smooth the tree using principal curves, and finally it projects each cell to its closest principal curve. So that's the technical language. All right, so this is a very well-studied task in single-cell RNA-seq analysis. And a downstream question is, once we have those trajectories,
we may be interested in, for each gene, how does its expression level change along the trajectory? Does it increase, decrease, or first increase and then decrease, right? We're interested in those questions because that can help us understand what genes are driving cells to change along each trajectory. So we call this differential gene expression analysis. And here are two example genes. Let's say that we already order cells by pseudotime, and the pseudotimes are normalized between 0 and 1. 0 means the earliest pseudotime, 1 means the last pseudotime. And here each dot is still a cell. And we are visualizing the expression level of this gene CCL5, whose levels are on the y-axis. So we can see that for this gene, clearly along the pseudotime, the expression level increases. And we hope to identify this gene as DE, differentially expressed. On the right-hand side is another gene. We look at its expression levels and we see it's almost flat, no obvious changes along the pseudotime. So we want to call it not DE. This is our goal, but what method can we use to achieve this goal? So to achieve this goal, we need a statistical method. All right, and before our work, there was a paper method called trace-seq. It does the job. It tries to test if a gene is DE based on a generalized additive model, which I will introduce briefly later. So this is short as GAN. And another method, Monaco 3, it is mainly for pseudotime inference, for finding the trajectories, but it also has a functionality to do DE. And this one uses a generalized linear model, not a generalized additive model. So I will talk about these two in the next few slides. And this is short as GLM. So the main difference between GAN and GLM is that GAN is more flexible by allowing the relationship, which is assumed to be linear in GLM, to be not linear. So it's more flexible and can capture more complex patterns. So both methods are regression based. So just for your information, both GAN and GLM are considered regression models. So in the regression model, we will have a response and a predictor so that the model will find a relationship. So we can use the predictor, or sometimes we call it covariate, to predict the response. So in our setting, the response is one gene's expression level in a cell, and the predictor is that cell's pseudotype. So coming back here, this is the predictor and this is the response. We want to use regression to fit a relationship. So you can think of the blue line as the fitted regression line. And the reason we want to develop a new method is that we realized the limitations with these two existing methods. So in particular, their main limitation is that both methods treat the cell pseudotime as observed. In other words, they didn't consider the uncertainty that's involved in the pseudotime inference step. As I said in the beginning, pseudotime is not observed, but inferred from the same data. And this inference itself involves uncertainty. So by uncertainty, I mean that even the set of cells, if I just randomly subsample a set of cells and then apply pseudotime inference, for example, by slingshot to that subset, then these cells may receive different pseudotime from what they have when I use other cells. So in other words, that subsampling giving me different pseudotime for the same cells imply the uncertainty in the pseudotime inference step. Okay, so our argument is that because these methods ignore this uncertainty, then their DE result, which are encoded as p-values. So we will have one p-value per gene. And intuitively, we know that a smaller p-value means a stronger DE effect, right? So we hypothesis that this modeling modeling may result in invalid p-values, and we want to address that. Okay, so here is some example to show the uncertainty, right? We have the cells, and this is the trajectory we inferred using all cells. But if we just use a subsample of cells, we can see that here still each row is a cell, and we just plot that cell's pseudotime across multiple subsamples. So each dot, they are very small dots, represent one subsample, and the cell pseudotime in that subsample. So we can see that it's a band, which means that this cell does not receive a fixed pseudotime across subsamples. Instead, its inferred pseudotime may have a range. That's what we mean by uncertainty. Okay, so with this, our proposal is PseudotimeDE, and how our method works is captured in this illustration. So we start from all cells, and we apply a pseudotime inference method such as slingshot to obtain the pseudotime. So you can see the cells are now ordered by pseudotime. And then we apply the GAN model, and I'll introduce this briefly later, to these cells for every gene J. For every gene J, we have one GAN model. And from this fitting, we can obtain a test statistic. And this is about testing whether the gene is DE or not along the pseudotime. And using the test statistic, we convert it to a p-value. Oh, sorry, we don't yet, we keep the test statistic. But you know, in that trait seek, we would directly go from test statistic to p-value. But we think it's problematic because here in this model, the pseudotime is considered as fixed instead of random. So to fix this issue, we employ subsampling. So you see that we subsample the cells into 3 subsamples here, just for illustration purpose. On each subsample, we apply the same pseudotime inference method to order the cells. And here, because what we need is to create a null distribution for the test statistic. That is, what the distribution of Sj would be like if the gene J is non-DE. So that null distribution is critical for our calculation of the p-value. All right. So for permutation, what we did is that we just randomly permuted the cells because we know that permutation can ensure the gene J is not DE since the cells are shuffled, right? So even no matter the gene J being DE or not, in the real scenario, after permutation, the gene J is for sure not DE. Then with the permuted subsamples, we fit the same model, GAN model. And then from each subsample, we can obtain a null value of this Sj. And let's say we have 1,000 subsamples, we will have 1,000 null values of Sj, and we treat that as our null distribution. And then we compare the true Sj, observed Sj, to the null distribution, and we will use the right tail probability of this null. That means the proportion of the null values here that are greater or equal than this value. And we call that our p-value. So essentially, we didn't use the theoretical null distribution of this Sj as is, like in that Tracy paper. So by theoretical null distribution, it's something you have learned from t-test, let's say. In t-test, the t-statistics null distribution is and that brief illustration or demonstration of what we mean by GAN. So in the GAN model, we assume the gene expression, okay, here is gene J in cell I. We call that it's a count number in single cell RNA-seq data. So we have a read count of gene J in cell I. We assume that this count follows a negative binomial distribution, and its expectation or mean is denoted by mu ij. And when we say DE, we're actually talking about the relationship between this mu ij, the expected expression, not the observed one, in relation to the pseudotime I, the pseudotime of cell I. So this is what we call the additive model, because basically here this function it's a nonlinear function of the pseudotime TI, go down here, is written as a summation of those cubic bases. So we call this in statistics, it's called cubic spline function. So in other words, each BK is a basis function. It is nonlinear, and we consider this Fj to be just a linear, just a summation of those nonlinear basis functions with coefficients beta jk for the kth basis. So that's why we call it additive model. And here the generalized means here the distribution is negative binomial, not normal distribution. Because if we have taken a linear regression class, we know that we often assume it's a normal distribution, right? So here's not. Yeah, so that's all I want to say briefly about this model. So quickly to take a look at the result. Oh, by the way, in our method, we consider two distributions. One is negative binomial, one is zero inflated negative binomial. So in the zero inflated negative, we consider that the count Yj may have more zeros than what we would expect under negative binomial. So we use this to account for the known sparsity in single cell data. That is, we have more zeros than we expect. So this is the fitted regression line. Basically, the line is showing this relationship, this beta J0 plus FJTI. And we can see that the red one is what we use by using the zero inflated negative binomial, while the blue one is just using negative binomial. You can see the difference is that using the negative binomial only, the line will be drawn down by the zeros, while if we use zero inflated negative binomial, the line is less affected by zeros. So we leave both options open to our users. The default is negative binomial, but users can specify the argument in our R function to be zero inflated negative binomial. The reason is that we think it's context specific, whether a biologist would think those zeros are biologically meaningful or not. If they think it's biologically meaningful, the zeros, they should use the NB, the blue one. Otherwise, they can use the red one. So the regression is less affected by zeros. Okay, so I'm going to skip that detail and let's look at the result. So the first one we are going to look at is this application to a data set. And this data set is about dendritic cells stimulated by LPS. So it's in the p-value distribution histogram. So we know that if the p-values are valid, then we should have the p-values uniformly distributed between 0 and 1. So except the genes which are DE that should receive small p-values, for other genes, the p-value distribution should be flat. And that's the case for PseudotimeDE, but not the case for TradeSeq. You see the tail here near 1, or the mode here near 1 is unexpected. It shouldn't be there for valid p-values. And Monaco 3, another method, has the correct p-value pattern. However, because it uses the generalized linear model, which is less than us, which means that it identifies fewer DE genes than we do. So basically what we do after checking the p-value is that we look at the DE genes found by us, found by TradeSeq, and found by Monaco 3. We do this Venn diagram to check the overlap. And we do see that TradeSeq is almost included in ours. So you see very few genes are identified by TradeSeq only, but Monaco 3 has some differences with us. So therefore, what we do is that we look at the genes that are only identified by us or by TradeSeq or by Monaco 3, and we check the gene ontology terms. This is called the functional analysis. We check what biological functions are enriched in the DE gene set. And we found that there are more Go terms enriched in the DE genes that are found by PseudotimeDE only. So this actually verifies that the DE genes found by PseudotimeDE are biologically meaningful. So please note that on the left-hand side, we use slingshot for pseudotime inference for finding the trajectories. And on the right-hand side, we use monocle 3, another method for pseudotime inference. We just want to show that our method works well with either pseudotime inference method. So we don't require pseudotime inference method to be fixed. Instead, our method is adaptive to any pseudotime inference method. That's the message. Okay, and we also look at the GO terms specifically enriched in the DE genes found by us. And we see that they are very related to immune responses. So which means that they are biologically meaningful. This also verifies the meaningfulness of our DE genes. Okay, so for our students, if you are interested in knowing more about generalized additive models, there is a book written by Dr. Simon Woods, who also wrote the R package we used in our method. So that's all I want to say about the first part. And for the second part, given the time limit, I think I'll be quickly to just go over the big idea. So here our motivation is to design a more realistic simulator to help users benchmark different computational methods. As you know, single-cell RNA-seq is a really popular field. And for each task, let's say cell clustering, I would say more than dozens or even close to maybe 50 methods have been developed for this task. So therefore, as a user, you want to know which method I want to choose or which method I trust the most, right? So then therefore, you want to do some benchmark. However, we don't know which cell clusters are true because if we don't know truth from real data. So therefore, a computational simulator that can give you the truth along with the data will be necessary for doing such benchmarking. And another purpose of the simulator is that we want to allow users to vary cell number and vary sequencing depth so that they can look at the data under different experimental parameters and see whether the data are of good quality for their analysis. That can give them some idea to design the experiments before they do the experiments. Okay, so before our work, these simulators were proposed, but the reason we want to propose a new simulator is because we realized that none of the methods except this recent one, which we just recently added to our comparison, these existing methods cannot really capture gene correlations. In other words, they are assuming genes are independent, but that's not the case in biology. But this simulator, the most recent one we found, although it can capture gene correlations, but it does not allow varying the sequencing depth. So that's why we think that it's not as flexible as our goal. So here's our proposal, SC Design 2. And the reason we call it 2 is because we have SC Design as an earlier simulator we developed. But in that simulator, we assume genes to be independent. So that's the thing we want to fix. So we have real data as input. So here we divide up, this is a gene by cell matrix. So rows are genes, columns are cells. We divide the matrix into K submatrices by cell type. So that means we require the cells to be either annotated as cell types or clustered as K cell clusters before inputting it into our simulator. So for each submatrix, we are going to estimate a joint distribution of all genes. So in our statistical formulation, we are treating the cells, each column here, as observations or data points. And we consider the genes, the rows, as variables. So let's say we have 20,000 genes, then we have 20,000 variables. And this joint distribution is the joint distribution of those gene variables. Then this distribution itself should preserve gene correlations. And then given this distribution, we can generate new cells as new data points or observations from this distribution. So for example, if we set the sequencing depth to be low, then our simulated cells or synthetic cells will look like this. You can see that compared to the real cells, the colors are more red, right? That here, the reddish means it's the counts are low, while the green color means the counts are high. So we can actually make the data correspond to the lower sequencing depth. And we can also change the number of cells. So the matrices we obtain can have the number of cells as the columns you specify. Okay, so using those data, our synthetic data can be used in whatever ways the users want to play with for benchmarking computational methods or to evaluate whether the sequencing depth and the number of cells are satisfactory. Okay, so again, I'm going to leave out the technical details, but I want to say that the key technique we used is what we call the copula framework. So the copula framework is a very interesting and powerful technique for us to integrate marginal distributions here, marginal count distributions, one per gene, into a joint distributions for all genes together. So using this framework, we are able to preserve gene correlations. That's the key. And so, as I said, I can share the slides with you guys later, but given the time limit, I'm going to skip the technical detail, but I'll provide a reference. So basically what we did is that given the input data, one matrix per cell type, we will fit a joint distribution for each cell type. And then given user specified input, including the number of cells and the total sequencing depth, we are going to output the synthetic data for each cell type. And we can also give users our fitted model parameters, including each gene's mean expression in each cell, each gene's variance in each cell type, and also the gene-gene correlations from each cell type. So users can use those data for their other analysis. For example, the gene-gene correlations for each cell type may be used for constructing a gene correlation network. And that's also a very useful analysis in bioinformatics. So I'm just going to show some results to see that we're really doing well for preserving gene correlations. Here we look at two correlations. One is Peterson correlation, the most common one. The other is Kendall's tau. It's a rank-based correlation that's more robust to outliers. And here we're plotting the violin plot as the distribution of all gene pairs correlations. So this is from real data. This is from our synthetic data. And this is our synthetic data. If we drop the copula step from our own model, we can see that the correlations are not preserved. And these are from other existing simulators. And we can see that even though some simulators may have been better, like this ZIMWave, SparSim, SCGAM, and this one is a deep learning based framework. So although they are theoretically possible to preserve gene correlations, but it's not really emphasized in their method, by the way. However, we see that like ZIMWave and SparSim seem to miss a lot of negative correlations, while SCGAM as a deep learning method is really slow to train, and also its distribution do not really resemble the real data as our distribution does. So this is the overall summary. And here is some graphical illustration of the gene-gene correlation matrix as a heat map. So you see here, each entry is for one gene pair. And we see that this is the data for training our model. This is the leave-out test data that are not touched. We see that our gene correlation matrix resemble the real test data the most. While without copula, we cannot do the job. And ZIMWave, SparSim, their matrices do not look like real data. This is for one type of single cell protocol, experimental protocol called SmartSeq2. Okay, and also we visualize the cells using the t-SNE plots, which are two-dimensional visualizations of cells. So here are training cells, test cells, synthetic cells. I think what's most informative is that we mix our synthetic cells with the test cells and look at them together. So here you see that the red ones are our simulated cells. The black dots are real test cells. So if the two sets of cells really resemble each other, then we should see the red dots and the black dots mix very well. And here, this MILISI is a quantitative index to indicate how well do the two groups of cells mix. The larger the index, the better the mixing. And we can clearly see that the mixing is the best for our assay design too, and not so good for other simulators. And furthermore, as I said, our method is applicable to other single-cell gene expression data, like MERFISH is a type of spatial transcriptomics data. So it can have cell spatial coordinates in addition to gene expression counts. But here we just try to simulate gene expression counts, and we see that we still do the best job. Okay, and finally, these are other applications. So here we vary the number of sequencing depth. We vary the sequencing depth from, say, 7.2 million to 457 million. And we can see that the synthetic cell look like this as we increase the sequencing depth. In particular, some cell types are better separated as the sequencing depth gets deeper. And indeed, we check the performance of SURAT and SC3 as two popular cell clustering methods. And we see that as we expected, both methods have better clustering accuracy as the cell sequencing depth increases. And also, we see a saturation effect for both methods, which is what we desire. So we know that once our sequencing depth reaches a point, then further increase on the sequencing depth will not lead to better accuracy for clustering. And we see that also by comparing the two methods, when the sequencing depth becomes really deep, then SC3 does better. However, SURAT is more robust for those low sequencing depth data sets. So it can still give some results, or it can still work for those data sets when the sequencing depth is really low, but SC3 fail on those cases. So we see this trade-off between accuracy and robustness. And on the right-hand side, we fix the sequencing depth, but we vary the cell numbers. So you see, we go from 400 cells to about 15,000 cells. And we want to see whether there is some trade-off in the middle, because we know that after we fix the total sequencing depth, if we sequence too many cells, then each cell will receive too few reads to be clearly captured. So in that case, the clustering will be hurt by the low per cell sequencing depth. On the other hand, on the other extreme, if the cell number is too small, then even though we have a very deep per cell sequencing depth, we do not have enough cells for each type to do good clustering. So the clustering accuracy will also be bad. And indeed, we see this trend in SURAT. So the clustering accuracy first increase and then decrease. So this suggests that there is an optimal cell number to sequence if we want to distinguish the cell types. And also here, the dashed vertical lines are marked for the sequencing depth in real data and the number of cells in real data. So these suggest that we may optimize our experimental design here by moving left a little to sequence fewer cells, and here by moving right a little to sequence deeper. That's the message. Okay, and also we can apply this for the task of detecting rare cell type. So here we consider two methods that can identify a rare cell type from single-cell RNA-seq data, FIRE and Jingyi class 2. And just to briefly summarize, our results shows that, again, for their best performances, Jingyi class 2 is better. However, FIRE is more robust as it can work for a wider range of cell numbers and a wider range of UMIs. So for example, here, the Jingyi class 2 is good overall when the sequencing depth is high. However, it has this unexpected drop here, which means that it had a mistake somehow, while for FIRE, we don't see that happen. So that also confirms the better robustness of fire. So at least you can see from these two cases that for computational methods, we cannot expect a method to be perfect or the best in every aspect. There's a method may be better in this aspect, but worse in other aspects. So as a user, I think computational benchmarking is really important to choose a method that you really trust. Okay, so if you want to know more about copula, this is also a book that gives a very good introduction. So finally, both methods, we provide our packages for you to try. And the key take-home message is the importance of some classic statistical techniques in our computational research. For example, the GAM, which is the 1990 method in the year 1990, and it's very useful for this task of finding DE genes along cell pseudotime. And for assay design 2, we use this copula technique to capture gene correlations. So finally, I want to thank my student Dongyuan for leading the PseudotimeDE project and Tianyi for leading this Assay Design 2 project, and my former student Vivian, who graduated from UCLA in 2019 and is currently an assistant professor of biostatistics at Rutgers University. And she also helped us with this Assay Design 2 project because she was the author of our older assay design method. And also I want to thank my funding agency for their support. Thank you.